Hello dear students. In last lecture we have discussed about the Koch postulate, Robert Koch, and how he defined the role of microorganisms in disease. And with respect to that, he have put a four rules, we can say, or postulates with respect to the role of microorganisms in the disease. Now, in this lecture, we will discuss about the major contribution of Sir Robert Koch in the field of microbiology and what are the different discoveries he has taken in his era, or what are the different contributions with respect to the field of microbiology. So firstly, he investigated the anthrax disease cycle. Normally the anthrax disease is caused by the Bacillus anthracis, and that disease cycle was invented or discovered or properly stated by the Sir Robert Koch in 1876. He studied the bacteria that causes tuberculosis in 1882 and cholera in 1883. So he was a well-known bacteriologist with respect to that. He also studied the bacteria which are responsible to cause a disease named as tuberculosis, and the positive agent of tuberculosis is nothing but a Mycobacterium tuberculosis in 1882, and the positive agent for the cholera, that is Vibrio cholera, was reported in 1883. He discovered the bacteria Anthrax bacilli, that is Bacillus anthracis, tubercle bacilli, and cholera bacilli. So with respect to the proper study of that positive agent of that particular different diseases were carried out by the Robert Koch. Koch observed the phenomenon of acquired immunity. He also observed the phenomenon of acquired immunity. It is nothing but a term which will be used in respect to the immunology. So acquired immunity is nothing but a, we can say it is acquired by the individual with respect to the particular infections. So with respect to time, different infection we have suffer, and in that response our body make a immunity, and that is called as acquired immunity. He introduced the solid media with the use of agar as solidifying agent. He also developed a pour plate method. So it is nothing but a well-known contribution with respect to microbiology, that is use of solidifying agent, and that is nothing but the agar. Normally we are also using that agar for solidifying our media in our laboratory practicals, or for the preparation of solid media. It is Petri plate. We also use, or it is normally a main ingredient which is used for the solidifying that particular media. So it is a agar that was used by the Robert Koch. He also developed the method for pour plate method. It is nothing but a pure culture isolation technique normally used in the field of microbiology with respect to how to isolate the pure culture. There are again two different methods, that is plate method and strip plate method. We can also use for the isolation of pure culture. Apart from that, pour culture method for the pure culture isolation was developed by the Sir Robert Koch. Koch also developed a media suitable for growing bacteria isolated from the body. The result was the development of nutrient broth and nutrient agar media. Still it is wide in use, or normally we have use that nutrient broth, that is liquid media, and nutrient agar as a solidifying or solid media for the isolation of bacteria, or to grow microorganisms on laboratory level. We can use that particular media. He also introduced method for isolation of bacteria in pure culture. So here we have taken that is pour plate method, or he also invented the different method to isolate how the pure culture from mixed culture. He described hanging drop method for testing motility. It is again another method we normally use in laboratory with respect to whether that organism having ability to show the motility. So for that we can use a cavity slide, and on that we put a broth culture of that particular organism, and by using that cavity slide we can check whether that organism is motile or not, and that method was given by Sir Robert Koch. He also invented the hot air oven and steam sterilizer. and also introduce method to find out the efficiency of antiseptics. So these are the again sterilization methods were given by the Sir Robert Koch, and how to check the efficiency of the antiseptics are also given by the Sir Robert Koch. So these are the major contributions in the field of microbiology, and which are also in the era of 18th century, you can say. So these are nothing but the major contribution of Robert Koch. Later on, Charles Chambers and they, with respect to that, different discovery and positive organism with respect to different diseases are we have taken here. The discovery of viruses and their role in diseases was made possible when Charles Chambers constructed a porcelain bacteriological filter. So after that, the proper isolation of viruses can be carried out, and it was again a important discovery with respect to the isolation of viruses. As they are small in size, they could not be filtered out or separated with respect to bacteria and viruses. So by using that filter, we can use or can be capable to isolate or to culture viruses. Here are the table. Here in one column we can see different diseases, and in other column we have taken a different positive agent of that particular disease. So first is anthrax. It is again a disease caused by the Bacillus anthracis. All Bacillus anthracis is a positive agent of that disease, that is anthrax. Gonorrhea disease, or in common words we can call as a gurney, and the positive agent of gonorrhea is nothing but a Neisseria gonorrhoeae. Another disease is typhoid fever, or in Marathi or local common word we can say it is nothing but a Visham Jwar, and it is caused by the Salmonella typhi. It is a positive agent of that disease. Tuberculosis, or in common words we can say Kshay Rog. It is caused by the Mycobacterium tuberculosis. Cholera, in common words we can call as a Patki. And positive agent for the disease is nothing but a Vibrio cholera. Diphtheria, मराठी मध्ये किंवा अदर वर्ड्स कॉमन लँग्वेज मध्ये आपण त्याला म्हणतो घटसर्प, and it is caused by the Corynebacterium diphtheria. It is a positive agent of that disease. Another is a tetanus, मराठी मध्ये आपण किंवा कॉमन लँग्वेज मध्ये आपण त्याला म्हणतो धनुर्वात, and positive agent for tetanus is nothing but a Clostridium tetani. Pneumonia, positive agent for the pneumonia is nothing but a Diplococcus pneumoniae. मेनिंजायटिस ज्याला आपण म्हणतो मेंदूचा ताप किंवा मेंदूज्वर त्याला म्हणतो आपण नायझेरिया मेनिंजायटिस पॉझिटिव्ह एजंट. प्लेग, इट इज नाउ इरेडिकेटेड कॉज बाय द येर्सिनिया पेस्टिस. बोटुलिझम, त्याला अन्न किंवा फूड पॉइझनिंग पॉइझनिंग ऑर्गॅनिझम आहे बोटुलिझम, किंवा फूड बॉर्न इन्फेक्शन आहे. तर बोटुलिझम इट इज कॉज बाय द क्लोस्ट्रिडियम बोटुलिनिझम. डिसेंट्री, dysentery is caused by the Shigella. Dysentery is a positive agent of that organism. Syphilis, it is again a venereal disease caused by the Treponema pallidum. And whooping cough, ज्याला आपण मराठीमध्ये डांग्या खोकला, तो Bordetella pertussis. So these are the different diseases which are discussed here, and these are the different positive agents of that diseases. Now we will discuss about the discovery of microbes or microbial effect on organic and inorganic matter. How we have already discussed either microbes are responsible to cause different types of diseases, and we have already discussed the different positive agent of different diseases. So again we have to discuss about whether microbes are involved in organic and inorganic matter. How they convert, or either they are involved in the conversion of organic matter to inorganic or inorganic matter to organic. So we have to discuss the two of the pioneers, that is Winogradsky and Beijerinck, made many contributions in the field of microbiology. So when we talk about the soil microbiology, so these two names are commonly we can or we have to discuss, as they have major contribution in the field of soil microbiology. Beijerinck and Winogradsky investigated the biochemical role bacteria play in the important natural processes, and which are that including nitrification, nitrogen fixation, and sulfur oxidation. So these are the commonly carried out processes in the soil, and they are discovered or investigated by these two scientists, and these are the most important processes in which atmospheric nitrogen can be fixed by means of different and make available for the plants use, and again their conversion into different forms. They discovered the bacteria which can oxidize iron, sulfur and ammonia to obtain energy, and many bacteria could incorporate carbon dioxide into organic matter like photosynthetic organisms. So they also observe or discuss the iron oxidizing, sulfur oxidizing, ammonia oxidizing bacteria, and they utilize their source of energy. And apart from that, also investigated the organism which use carbon dioxide for their energy, like photosynthetic organism, and all these details are given by that two scientists. Iron oxidizing bacteria are non-harmful bacteria that produce their energy by oxidizing the iron, that is Fe2 plus and Fe3 plus ions, that is naturally present in subsoil. So these are discussed by that scientists, and they are nothing but the iron oxidizing in the case of iron oxidizing bacteria. The common sulfur oxidizing bacteria is Thiobacillus. Thioxidans is a chemolithotrop utilizing thiosulfate and sulfide as a source of energy to produce sulfuric acid. And all these details, or we can say in sulfur oxidation, Thiobacillus thioxidans are also involved in the oxidation of sulfur. So all these are mechanism. Microbes are involved in conversion of these simple inorganic and organic forms of different organic matter. Microorganisms accomplish major element transformation with respect to mineralization and immobilization, and they are normally carried out with respect to the soil. So here are two different methods, that is mineralization and immobilization. So here they convert the complex organic nutrient into simpler inorganic compound, and that process is called as mineralization. So here we can say normally in soil or everywhere some substances are produced by, like we can say they are complex in nature, and they are converted into simple inorganic substances by the activity of different microorganisms, and that process, that is conversion of complex organic substances into inorganic substances, simple inorganic substances, is called as mineralization. Apart from that, it is again so that plants can make use of that simple substances, nutrient, and these microbes absorb the simple minerals which are converted by the process of mineralization and prevent them from leaching out, and thus conserve the essential nutrient in the soil. So again, that simple conversion are absorbed by the plant root or make available by the microbes or the plants use, and they again convert that simple inorganic substances into complex organic substances by means of plants, and that process is called as immobilization. So both the cases, in case of mineralization and immobilization, microbes play a very important role in that with respect to that. So that when they die and become a part of the organic matter, these essential nutrients are once again mineralized by the microorganisms for plant use. So after the completion of the complete cycle of plants, what happens? The dead body or any organic rich substance are now converted into simple inorganic form by means of that microorganisms, and that process is again called as mineralization. So where complex organic substances are now converted into simple inorganic substances and again make available for the absorption of plants. So these are nothing but the process of mineralization and immobilization, and all these processes microbes play a very crucial role in all these two processes. Thus the soil fertility that is created by the microbes is also conserved by the cell. So normally we have discussed that is nothing but the role of microbes in conversion of or effect of microbes on organic and inorganic substances. So these cycles are naturally carried out in soil where complex substances are converted into simpler form, and simple substances are again converted into complex form, and this cycle is continuously carried out. After the completion of cycle of the plant, again by the process of decomposition or you can say mineralization, that complex substances are again made available or mixed with the soil and again made available for the as a source of nutrition for the plants, and in that way soil fertility can be maintained by means of that microbe. So these are the nothing but the major role of microbes with respect to organic and inorganic substances. Next lecture we will discuss about the recognition of microbial role in case of fermentation. We will discuss in next lecture.